In part 1 of this series, we discussed the nature of genetics, paying particular attention to how complicated it is. In a nutshell, you need a ton of information about what many genes are doing at once to really understand genetics. But for many decades, we were stuck with the plotting one gene at a time approach. Genomics allows us to analyze the behavior of thousands of genes at once, which is why it's been so transformative for the field. In this second video, we'll talk in more concrete terms about genomics itself, narrowing down our focus to a single topic, genome assembly. This is the initial step in most genomics work, and the quality of an assembly is foundational for most downstream analysis. Let's start our discussion of genome assembly by walking through the simplest workflow for how to do a genome. First, the scientist extracts and purifies all the DNA from their organism of choice. Then you prepare the DNA for sequencing. How this works depends on the machine you're using for sequencing, since the tech varies, but usually after extraction you break up the DNA into pieces of the right size. Next, you make what is called a library, which refers to manipulating the pieces of DNA such that the machine can sequence them. This often means attaching little adapter pieces of DNA to each side of the genome fragments so that the machine can bind them for sequencing. If this is too fast, by the way, you can pause and look at the graphic. After sequencing, the scientist gets back a huge text file with the sequenced reads. Here's a read from the Illumina machine. Each read basically has a name, some identifiers, and the sequence, which is just a series of A's, T's, G's, and C's. Sometimes there are N's as well, which means that the base at that position was not identified. The whole file from the sequencing run is millions or even billions of these reads, and they span the whole genome many times over. Basically a genome broken up into an enormous number of overlapping fragments. Once you have all your reads, you must then assemble them back into the genome. Remember, we started out with a small number of very large DNA molecules, the chromosomes, which we broke into pieces. Ideally, we would like to reassemble the reads, the pieces, back into the chromosome length sequences, but this can be quite difficult. To see why, let's review how assembly works. I introduced it in part 1, but it's actually a lot harder than I led you to believe. The basic mechanism, remember, is to look for regions of overlap between reads in order to align them and get back the original sequence. Let's illustrate it with a very simple example. Here we have one piece of DNA for which I don't know the sequence. Let's say I break it in 3 pieces, now called reads, and I get the sequences for them. Here you can see how the regions of overlap, in orange for reads 1 and 2, and green for reads 2 and 3, can be used to put the 3 pieces back together. So in principle, assembly is simple. I now need to explain why it's hard in practice. It's basically because DNA contains a lot of repetitive sequence. We might have AA, T, G, G, for example, repeated over and over again, thousands or even millions of times. Repetitive DNA like this used to be called junk DNA, by the way, but that's actually not a good name, since the mechanism leading to these repetitive regions is important in evolution. But that's beyond the scope of the present video. We're rather interested in the effect of such repetitive DNA on assembly. To explore this, let's suppose I break up a piece of DNA into 3 pieces again. In this example, both reads 2 and 3 have the same overlap, shown in blue, with read 1, meaning I can't tell the order of the 3 reads in the original DNA sequence. The overlap here would be caused usually by one of the many repetitive sequences scattered across the genome. And simple repeats aren't the only problem. Another issue is that many genes have common elements. Genes are made up of exons and introns. The exons contain the sequence used to make proteins, while introns are clipped out of the mRNA before the proteins are made. Say we have 2 genes, genes A and B. Further, suppose that each gene has 5 exons, and 2 of them are the same, colored red here, while the other 3 exons are different. That means that reads from gene A will overlap with reads from gene B, even if the 2 genes are on separate chromosomes. Genes with common exons like this, by the way, are said to be in the same family, and gene families can have hundreds of members. Worse still, they're sometimes in long rows in the same chromosome, making assembly quite difficult. I won't go any deeper, but what we've seen thus far should make it clear that although the principle of genome assembly is easy, the logistics of doing it can be difficult, or even impossible in some cases. Now we get to a key aspect of the problem, which is read length. There are 2 kinds of sequencing technology, short read and long read. Short reads are on the order of a couple hundred bases long, while long reads can be many tens of thousands of bases long, even up to 100,000 bases. Let's cut to the chase. Short reads are obviously far worse for assembly relative to long reads because they can't span repetitive regions. Nevertheless, with short reads alone at present, you can get a genome of sufficient quality to do many important genetic analyses. Ultimately, however, the overall quality of a genome assembled with short reads only is far less than we would like. There is a caveat, though. Bacteria and viruses have little repetitive DNA, and their gene families are small. Hence, a high quality genome assembly with short reads only is possible for them. It's for multicellular organisms, plants and animals, that the problems of repetitive DNA and closely related genes make assembly a nightmare with short reads alone. This is important because in the early days of genomics, we basically only had short reads. Long read tech took a long time to become good enough and cheap enough to use on a wide scale. So many mediocre genomes were assembled for plants and animals with short read tech alone for a period of about 10 years. Fortunately, however, long read tech has massively improved in the last 5 years or so and finally taken over from short read tech for genome assembly. In time, those suboptimal genomes made with short reads alone will all be fixed. I should say finally that assembly is even harder than I have made it out to be so far, because of the fact that many organisms have 2 copies of each chromosome that vary. But I think we've covered it in sufficient depth for an introductory video. In sum, and this is really all you need to know, we can now sequence a typical animal or plant genome for maybe a few $1,000 with really good accuracy. We should talk a little now about what a genome actually looks like. The genome file that results from assembly is just a big text file. You might suppose, for example, that if a species has 5 chromosomes, then our genome file should have 5 long sequences of DNA, one for each chromosome. But that is not the case. Instead, we have to talk about contigs and scaffolds. A contig is a piece of DNA for which we have a complete assembly. In other words, we were able to completely assemble that part of the genome from the reads. Contigs are arranged into what are called scaffolds. A scaffold just means that we know that some contigs are on the same chromosome, and they are in a particular order, but there is repetitive sequence of unknown length in between them. With the best sequencing technology and software, a genome has a scaffold number close to or about the same as the chromosome number, but each scaffold has many contigs, probably 50 at a minimum, and often many hundred. If I went too fast here, pause the video and study the graphic or watch this section again. Well, I think that's enough for one video. New science is a lot to take in. In the next video, we'll cover annotation, which is how we identify the genes and other important parts of the genome. We'll also introduce RNA-seq, which is one of the more important applications of genomics. If you benefit from or enjoy this content, then please like and subscribe. See you next time.